So, tough title. So I'll try to give you introduction about why glycans are important and what kind of evidence we have for the importance of glycans. Because unfortunately, the most frequent question I hear is, what are glycans doing at all? Are they important? They seem to be irrelevant to most of the scientific community because they simply ignore it. But before I start, I have to make a disclaimer. So many people think I'm a glyco fanatic because I think glycans are the most important. So maybe the fact that I publish papers like Glycans, the Third Revolution in Evolution don't help this view. But I do agree that the proteins in DNA are also important. I do think that proteins in DNA do something. But everybody knows that. So there's no need to persuade other people that DNA and proteins are important. But from a mysterious reason, some people think that glycans are not important. So it is important to remind them that glycans are also important. And through most of my talk, I will be talking about the importance of glycans. So the fact that I talk about the importance of glycans does not mean that the proteins in DNA are irrelevant. It just means that glycans are equally relevant. So when you think about the protein, you have to think about the glycoprotein, because actually the vast majority of proteins are glycoproteins. So in addition to the polypeptide part, which is presented in gray in this figure, there are colored parts which are equally important elements of structure. So structure which performs a function is a combination of a polypeptide, which is defined by a gene, and a glycan, which is defined by a complex network of genes and environment and everything else which affects protein glycosylation. And similarly to what Anne has showed, when you look at the cell surface, these few nanometers here, this white band, this is a plasma membrane, which we all learned about in high school and then at university, how important it is. And it is covered by this thick layer of glycans, which is 20, 30, 50 times thicker than the plasma membrane. So anything talking to the cell actually has to talk to the glycans first, and then it can talk to the proteins and everything else on the membrane. So when you think about it, my view of glycans is that glycans are kind of a coating we can put on ourselves. So if it is cold, we can have glycans which will adapt us to the cold weather. But when it is warm, we can change the glycans and be better adapted to the environment. And occasionally, we can make a mistake. We can have glycans which are wrong for the current situation that we are in. So we will be less fit for the environment if we have this type of glycans than if we have this type of glycans. And when we think about the cell surface, it's more like this. We have a thick protective walls of glycans which are between us and the environment, and they decide our future. And they're not encoded in our DNA, at least not directly. So each individual protein has its gene, which determines the structure. So the amino acid sequence of our proteins is determined by our genes, which we inherited from our parents, they inherited from their parents, and the sequence of a protein changes very slowly by random mutations. Glycans are not hardwired in a genome. We can change our glycans by simple change in the gene expression, intracellular localization, metabolite concentrations. We can change the glycans we make and make a different structure to perform a different function in a different environment. And this is all done in a very complex network of hundreds of genes which are regulated with thousands of different transcription factors and all other molecules, which then ends up in a final structure. We'll have a talk about this by Professor Kelly Moorman on Friday, so you will learn much more about it from him than from me. I will just tell you that this results in something like at least 2,000 different glycan blocks, glycan determinants, which can be created. And these glycan blocks are added to proteins to compose the final glycoprotein. And when you talk about the glycoproteome, which is all glycoproteins in our body, it is at least several orders of magnitude more complex than the proteome. So we talk about 22,000 genes, maybe hundreds of few hundred thousand proteins, and then we talk about millions or hundreds of millions of different glycoproteins which enable us to function the way we function now. And we know glycans are important. I believe most of the people in this conference believe glycans are important. And fortunately, other people are starting to think that too. We had a very strong policy paper endorsed by the US National Academies, which clearly stated that glycans are directly involved in the pathophysiology of every major disease. And additional knowledge from Glycoscience will be needed to realize the goals of personalized medicine, and personalized medicine is something which is a priority all around the globe, and to take advantage of the substantial investments in the human genome and proteome research and its impact of human health. So what do glycans do? In principle, and this is just my view, so this is not a generally accepted approach, glycans have 4 modes of action. One is they're ligands for specific glycoreceptors, often called lectins. So glycans are ligands, lectins are receptors, they bind to each other, we have a recognition, and then we have some kind of event after that. Selectins, pathogens, these are the examples. I will come back to that. The other option is, did you do alternative glycosylation, meaning you attach different glycans to different membrane receptors? And in this way, you modulate membrane dynamics. You decide for how long will a specific receptor stay on a cell surface. This is extremely important for receptor activity, and it seems to be regulated by alternative glycosylation. A similar thing is that by doing alternative glycosylation of any protein, you modulate its structure, and then you modulate its interaction with the specific receptor. And I will show some examples later. And then as a 4 model, we have this glycosylation as a simple regulatory modification, like an O-glucnAC. You put an O-glucnAC to change protein function or to prevent a change by phosphorylation. So let's go through these 4 examples. So at the cell surface, nearly everything is glycan. So anything approaching the cell, being it the pathogen like the viruses or the bacteria, or any other cell in our body, or even any other molecule in our body, it has to talk to glycans. And one of the best examples is something what Anne already mentioned, are the selectins. So the selectins are the proteins which bind to glycans, and this is the primary signal of inflammation. So if there is something wrong going in our tissue, there are cytokines signaling to the cell membrane, which present different glycan structures or different selectins. They bind together, neutrophils start to roll, then there is a hard attachment, and the neutrophils go in and kill everything they need to kill. So recognition between a lectin and a glycan receptor performs specific function. But the limit to this is the need of protein receptor. And to have a protein receptor, you have to have a gene. So you cannot change this process without changing genes. So this is just one of aspects how glycan function. Much more variable way how glycans can function is to put the different sugars on a protein and then to have a different function directly, without the need to change a DNA to have a novel function. For example, practically all membrane receptors and the membrane channels are glycosylated. And the glycosylation either directly modulates their activity or modulates the half-life of these receptors on the cell membrane and modulate activity in this way. And this is being done actually in a very this has been regulated in a very complex way, because you have a genetic pathway to synthesize a glycan. So any sequence variants in any of these genes will result in a little bit different pattern of glycosylation, of different ratios of different glycans. Also, epigenetic regulation of each of these genes will have a similar effect. So epigenetic downregulation of this gene could have exactly the same effect as a mutation which is making this gene less active. So genetics, which is our long history in thousands and millions of years, epigenetic, which can be our immediate history, just remember to the epigenetic modification, but also our current history, which is the sugar nucleotides which are needed to glycosylate. So hexosamine pathway overflows into the sugar nucleotide pathway. So depending on our energy state, we'll have a different concentrations of UDP-glycanol other sugars. So it's a long-term past, short-term past, presence, integrating into the decision how to glycosylate the protein, and then eventually